Good morning. Welcome to the BMES Professional Development Webinar, Connecting Leaders in BME with Underrepresented Groups. I am Guillermo Amir, and I will be your moderator today. This webinar is intended to be part of a series organized by the BMES Diversity Committee to give BMES members that are part of underrepresented groups a chance to hear from and interact with leaders in the field and provide a means to potentially forge new relationships. I am a professor in the Biomedical Engineering Department here at Northwestern University. I'm also affiliated with the Department of Surgery at Feinberg. And it's a pleasure to be here and to have our first guest speaker, Professor Robert Langer. Before I get started, there are a few things that we have to take care of, a few housekeeping items. First, this webinar is being recorded for your convenience. The slides and archived recording will be available in the next several days through the BMES webinar and the e-learning page. Next, at any time during the webinar, it is possible that you can submit questions, time, chat box, or message. Please submit your questions as we go along. The questions will be addressed by the speaker at the end of the webinar. And lastly, please do not exit the webinar right at the end because there will be a brief evaluation. I'd like to get your feedback, and we greatly appreciate that you answer this if possible. So please take a moment to complete it. There are a few polling questions that I'd like to ask to get a sense of your background, the audience, and for future directions. To answer the polling question, select your answer and click Submit directly on your computer screen. Please take about 30 to 40 seconds to answer each question, and I'm about to read them all out loud for you. First question, which of the following best describes you? Undergraduate student, graduate student, postdoc fellow, pre-tenure faculty, post-tenure faculty, or other? Good, so we have quite a bit of graduate students and some post-tenure faculty. Great, so about half of you are graduate students, a third of you are faculty. Next question, what is your ethnicity? All right. So, very good. We have a nice distribution of folks. Very nice. And the third question, what career path are you in or are you considering to pursue? We've got academic research, industry, entrepreneurship, business, other, none of the above. All right, wow, about half of you are in industry, half of you in academic research or want to pursue that. A few of you want to be entrepreneurs or are in that area. So this is a very, very nice distribution. So again, welcome everybody. And now, without ado, I'd like to introduce our inaugural speaker. And I'm very proud to be part of this. He was my former former PhD advisor and still a friend today, many years later. So Robert S. Langer, he's the David H. Koch Institute Professor, and there are 13 Institute Professors at MIT. Okay, and this is the highest honor that can be achieved or awarded to a faculty member. He has written over 1,350 articles, and he has over 1,100 issued and pending patents worldwide. Dr. Langer's patents have been licensed or sub-licensed to over 300 pharmaceutical, chemical, biotechnology, and medical device companies. He is the most cited engineer in history. His H-index, as a reference, so we all know what we have to aspire to, is 229. So this is great. So he served as a member of the United States Food and Drug Administration Science Board, the FDA's highest advisory board, from 1995 to 2002. And he was chair of that board from 1999 to 2002. He's received many major awards, over 200 of these. One of 4 living individuals to have received both the United States National Medal of Science and the United States National Medal of Technology and Innovation back in 2011. He also received in 2002 Charles Stark Draper Prize, considered equivalent of the Nobel Prize for Engineers. The 2008 Millennium Prize, the world's largest technology prize, the 2012 Priestley Medal, the highest award of the American Chemical Society, and in 2013, the Wolf Prize in Chemistry. So I would like to go on and on, but by the time I'm done, the time for this webinar would be pretty much over, so I'm going to cut that short. What I would like to note is that Forbes magazine back in 1999 and the BioWorld back in 1990 named Professor Langer as one of the 25 most important individuals in biotechnology in the world, an achievement that I and many of his friends and former students and postdocs are very proud of. So without ado, Bob, I want to turn this over to you so that we can go ahead and get started. Thank you very much once again for being our inaugural speaker.
Thank you, Guillermo. Well, it's a pleasure to speak to you. I thought I would tell you a little bit about my career and what we're doing, and then make some comments addressed to underrepresented groups and answer a few questions. So just a little bit about me. I'm a chemical engineer. I got my degree in 1974. And when I got done, the most common thing for a chemical engineer to do was go and work in an oil company. They had lots of jobs. There were these huge waiting lines at the gas stations. And so I thought that's what I would do because that's what all my friends did. And I actually got about 20 job interviews, 4 from Exxon alone. But I wasn't excited about that. I felt that I was looking for some way that I could use my chemical engineering background to make an impact on people's lives. And so at any rate, I started to think about what could I do that would enable me to do that. And one of the things that I did when I was a graduate student was to help start a school for poor kids in Cambridge. Cambridge, which has Harvard and MIT, so you might think that that would be a very rich or wealthy city with lots of advantaged children, but it turns out that in the 1970s, Cambridge had the highest high school dropout rate of any city its size in the United States. And I got involved because some people asked me about helping get a school called the Group School started, where I would help create the math and science programs. And they actually made a little bit of a video. Some of it's in the Chemical Heritage Foundation and other places. So I was going to start and just show you a little bit about that. And we're going to put that on.
It also offered Robert a transformational teaching experience at an alternative high school nearby called the Group School. The Group School was established primarily to serve students from working class and low-income backgrounds. It operated under democratic school principles, an educational ideal in which democracy is both a goal and a method of instruction.
The school was aimed at trying to help those kids who had dropped out get back and learn things, and I was very successful. I mean, I think the people working there were very dedicated. My job was actually, I was in charge of the math department and the science department. That was very, important time in my life. Basically, I probably do 2 things. I worked in the lab and I worked at that school.
At the Group School, Robert worked to develop a math and chemistry curriculum for high school students that was fun and academically solid. At MIT, he pursued his graduate work, doing different kinds of experiments aimed at ATP regeneration.
So anyhow, that was a wonderful experience for me. And then one day, I actually happened to see an ad in Chemical and Engineering News to be an assistant professor at City College of New York to develop new chemistry curriculum. And I was really excited about that, so I wrote them a job offer to see if I could be an assistant professor there, but they didn't write me back. But I really liked the idea, so I found all the ads I could to be assistant professor to develop new chemistry curriculum, and none of them wrote me back. So at any rate, that wasn't working so well, so I started to think, what other ways could I use my chemical engineering education to help people? And I thought about medicine. So I wrote to a lot of hospitals and medical schools, and they also didn't write me back. And then one day, one of the postdocs in the lab where I was said to me, Bob, he said, there's a surgeon named Judah Folkman at Boston Children's Hospital, and they said sometimes he hires unusual people. So he thought very highly of Dr. Folkman. I don't know what he thought about me, but anyhow, I wrote Dr. Folkman, and Dr. Folkman was kind enough to offer me a position. And I'm going to show you what we were trying to do on the first slide. This is actually from the New York Times, I think in 1971. And it was Dr. Folkman's vision of how tumors grew in relation to their blood vessels. And his idea was that a tumor would start out very small, and then it would start to grow and multiply without control. But one of the problems it ran into is a chemical engineering problem, a nutrition problem, that cells in the center would die because they can't get nutrients or get rid of waste. And so they couldn't grow further than this, except for the fact that what he said is that the tumors produced a substance which he called TAF, or tumor angiogenesis factor, and that caused the surrounding blood vessels, which normally didn't do anything, to start growing right to the tumor. And then the tumor would get bigger and bigger and bigger, and of course it could spread through those blood vessels, and that's a process called metastasis. And his thought was that would there be some way to stop the tumor from growing, and if you could, in other words, stop the blood vessels from growing, and if you could, then you could get hopefully a dormant tumor. Well, to try to solve a problem like that, I think to us required two things. First, we had to find a substance that could even stop blood vessels from growing, and actually the conventional wisdom at the time was that no such substance existed. In fact, I didn't realize this when I first went to work there, but almost nobody believed this theory. At any rate, we started, and one of the things we started looking at to see if we could find a substance that could stop blood vessels from growing was cartilage. And I went to, started out going to, looking at some of the little animals in the lab, but we couldn't get much cartilage there. So then I went to a slaughterhouse in Cambridge, and I got a cartilage bone. You see an example of that here. But still, one bone you can't do that much with. So I found out where do all the bones in the northeast United States go. And it turns out that they go to these meatpacking places in South Boston. So I made an arrangement with them to get all their bones, and I'd go there 3 to 4 times a week, bring them back, and then they had some meat on what you see as the white part of this, and I would scrape that off with what's called the periosteal elevator. That's a surgical instrument. And then that would make them look like this. That was actually the first step of the purification I devised. And then you slice this off, and then we would extract it with different solvents like guanidine hydrochloride, and then put it through different columns to purify it. And at the end of a couple years, we maybe had 50 to 100 different fractions that we wanted to test. Now that then brings up the second problem: is how would you test it? And if you look back at the history of medicine, one of the biggest problems in testing things is a bioassay, and we didn't really have one, so we had to invent one. And what we decided to use was the eye of a rabbit, and the eye of the rabbit the rabbit's eye doesn't normally have blood vessels. But what was discovered by Michael Gimbroni was that if you put a certain type of tumor in, it would cause blood vessels to grow. It took about 8 or 9 weeks, but that would actually happen. And the blood vessels would grow, and they would grow to the tumor. I'm going to show you that a little later. But what we thought is we could create an assay by using an ophthalmic microscope to measure the length of the longest blood vessel. And that could even be a quantitative way of looking at this. And then we would have a controlled release polymer that we could put next to it, and that would release anything that we would put in. And that was sort of the idea. Now, one of the challenges was that the molecules that we had isolated were large molecules. and no one had developed a way to release them before, and in fact, people didn't think you could do it, as I'll get into. So we also needed to make sure that they were non-inflammatory in the eye, and that was also a big challenge. But the biggest challenge was, could molecules slowly be released from them? Well, Dr. Folkman was on the board of the one company, Alza, that had done any work in this area, and he asked them, could they help us? And they said, well, no, you can't release large molecules. They said they just can't diffuse through solid polymers any more than one of us could walk through a wall. And the literature would say the same thing. It said, like, this is just one article, the use of polymer matrices for slow-release systems has been virtually restricted to small molecules. And sometimes what I say is the only reason we solved this, maybe, is that I hadn't read any of this. So I spent 2 years in the laboratory experimenting with different techniques, and I actually found over 200 different ways to get this to not work. But eventually, we were able to make little microspheres, and I'll show you one on the next slide, and one cut in half. And it turns out that you could use these to release molecules of any size. This is shown on the next slide. This was from a paper Dr. Folkman and I wrote in Nature in 1976. You could release them for over 100 days. And later on, using some chemical engineering approaches, we showed you could even get constant release. That's shown on the next slide. We actually did some mathematical models predicting how to get constant release, and then verified that. This was done by Bill Ryan and others, Dean Shea. At any rate, then, so I should say that, so we did this work, we published it, but it met with an enormous amount of skepticism. In fact, I remember when I was 27 years old, I gave my first talk at a scientific meeting, and I practiced this talk for hours, actually days, even weeks. And I was very nervous giving the talk. And I got up and I gave this talk, and I was by far the youngest person there. But I actually thought at the end of it I didn't do that badly. I didn't stammer too much or forget what I was going to say. So I thought when I was done with the talk that a lot of the older chemists and engineers, being nice people, would want to encourage me, this young guy. But when I got done, a whole bunch of them came up to me and they said, we don't believe anything you just said. They said that this approach can't work because the large molecules can't diffuse through solid polymers. Same thing the Alza people said. They also pointed out that we were using organic solvents, and they said that will destroy whatever we put in. So, and actually things got worse from there. They got worse. What happened is that I was working in a surgery lab, and a lot of my friends said, well, you shouldn't be a postdoc forever. You should apply for faculty positions. So I did, but and I applied to chemical engineering departments, and not a single one would hire me. And I actually also got my first 9 grants turned down as well. And then eventually I got a job in nutrition department. But what happened was, a year after I joined that department, the department head, Nevin Scrimshaw, who hired me, decided to leave. So a number of the senior faculty decided to give me advice, and their advice was that I should start looking for another job. In fact, on the next slide, one of my good friends, Mike Marletta at the time, he now is a chair at Berkeley. This was a talk he was giving when I won an award a few years ago. He was saying what it was like to be working with me back in the late 70s, early 80s, and this is just a direct quote that was from his talk. And he said, one evening he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for polymer drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. I thought I was in a Fellini movie. At any rate, so it was very discouraging, but I kept working on it, and fortunately for me, other people started using this work academically. But still, people were very puzzled how this worked. So what we did is we began doing fundamental studies. This was done by Rajan Bawal, one of our students, and he began studying the microstructure of these systems and to help figure out the mechanism. Now, the next slide shows a micrograph of one of those materials that doesn't have any drug in it. And if you tried to do a permeability study, a molecule couldn't diffuse from one side of this to the other. But if you put a drug in, and the next slide shows that, this is before any release, you kind of see what we call a phase separation. The polymer is the white part and the drug is the red part. And if you release this for over a year, then what happens is it looks very, very different. Left behind where the myoglobin was are these pores. And you'll see this on the next slide. And these pores are large enough so that molecules even 300 molecular weight can't get from one side to the other. I'm sorry, these pores are large enough so that molecules even 2 million molecular weight can get from one side to the other. But it turns out that if you do a lot of microstructural analysis of these pores, they have tight constrictions between them even though they're connected, and they're also very, very winding and tortuous. So it takes a really long time for the molecules to wind their way through. And in fact, over the years, a lot of our students, like Bill Ryan and Dean Shea and Larry Brown and Mark Saltzman and many others, Ron Siegel, what they've done is they've shown that and developed mathematical models to predict the porous structure in these, to better understand it, to lay out ground rules for how you can get release and control it anywhere from days to years. Well, since we did all this, it also put us in a position where we could now release these putative angiogenesis inhibitors. So I'm going to go back and show you a slide of the assay we wanted to use. And now we had the tumor and we had the polymer. And we did about 2,000 eyes. And it turns out that almost every one of the fractions that we tested did not work. And sometimes I kind of think of them as a control. And in fact, I think that's really almost appropriate because, right, they're isolated from the same substance. They're isolated even with the same extraction procedure. They're just different purification procedures. And almost every one didn't work out of many, many, but we found one that did. And I have to apologize in advance before I show you this slide if anybody doesn't like the sight of blood. So if you don't like it, don't look. But this will graphically show you what happens. On the left-hand panel, these are both at about 9 weeks. What you'll see is if you look closely, you'll see a tumor, and you'll even see the edge of the polymer. And this is without the cartilage-derived inhibitor. But what you see is that literally a sheet of blood vessels grow over the polymer on the way to the tumor. And if you looked at this eye just 3 weeks later, it would be three-dimensional, out of the orbit of the eye, and it would ultimately kill. In contrast, we sacrificed the animals before then. In contrast, when you put the inhibitor in, again, here's the tumor, here's the inhibitor. Look at how the blood vessels are lower, they're sparser, and they actually avoid the polymer. And this only happened with this inhibitor. And it was the first time we published this in Science in 1976. And it was the first time that it showed that you could inhibit blood vessels. But still, it took a long time before these could get used clinically. It just took millions, billions of dollars, actually, for many pharmaceutical companies like Genentech. And on the next slide, I want to show you what eventually has happened. And that is now some of these are the most common, most widely used drugs in history, in biotech history, for treating cancer and other diseases. Like Avastin is, for example, the #1 or 2 best-selling biotech drug in history. And not only can you treat many different kinds of cancer, but you can also treat different eye diseases like macular degeneration with drugs like Lucentis and Eylea. And so it's really led to all kinds of new therapies when there weren't any before. And the FDA even now says there's 4 ways to treat cancer: radiation, chemotherapy, surgery, and angiogenesis therapy. And so that's been nice to see that happen. Now, I wanted to also, it occurred to us that maybe this would be useful not only from the standpoint of isolating new drugs, potentially, but also in its own right, because so many drugs, have short lifetimes in the body and can't be given very easily. So we decided, this was done by Larry Brown for his thesis, could we deliver insulin just as a proof of principle? And the next slide shows a tiny little pellet that we put in, smaller than an aspirin pellet, slowly releasing insulin for over 100 days. And you could take the blood level from high to normal by doing that. Now, Dr. Folkman and I, one of the things that we've also been very involved in is kind of entrepreneurship. And I didn't start out that way, but I did want to see our work get used by pharmaceutical companies and by patients. And one of the things that Dr. Folkman said is that we should file a patent, and this was 1970s. Now, it's interesting. Today, that's very common, particularly for us. But at that time, Children's Hospital, one of the best children's hospitals in the world, never had a patent. So we filed for the first one, and 5 years in a row, 5 times in a row, it was rejected by the patent office. And the lawyer for the hospital said, we should just forget it. It's never going to be allowed. So at any rate, what I thought about was, as I mentioned to you earlier, a lot of people told me this would never work and it couldn't be done. And I wondered if anybody ever wrote it. And so wrote that down. So I did what's called a science citation search to see if anybody ever wrote about any of the things we did. And I did this in 1982, looking back to see who might have written about our 1976 Nature paper. And on the next slide, I want to show you one of the articles I found. And I had no idea this was written. It was written by 5 of the top polymer chemists in the world. And what they said, this is a direct quote, is that generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, Folkman and myself have reported some surprising results that clearly demonstrate the opposite. So what happened is I showed this to the lawyer. He was very excited, and then he flew down to the patent office to show it to the patent examiner. And the patent examiner said, gee, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from these 5 people that they really wrote that. So I wrote each of them, and they were all nice enough to write me back that they really wrote it, and we got a very broad patent. And and that's on the next slide. And with that patent, we began teaching different people how to do this. I got involved in starting companies. Today, actually, as Guillermo knows, I think there's over 30 companies that have been started from our lab, mostly by students in the lab. I've helped. And that's led to all kinds of products. Some of these are shown in the next slide. And they've affected tens of millions of patients. I want to close on a few things. First, I wanted to talk a little bit about underrepresented groups that I've had in my lab, and I've been very proud of how well the people have done. I just wanted to highlight 3 of them just on actually the last slide I'll show, and then I'm going to answer some questions. And I've had many, many underrepresented groups in our lab, but I just thought I would highlight these 3 to give a little bit of diversity on this. The first I wanted to mention is Cato Lorenzen. Cato actually has had an amazing career. He actually was Dean of Medicine at University of Connecticut. He's a chemical engineer and an MD, orthopedic surgeon. He's also Chief Executive Officer of the Connecticut Institute for Clinical and Translational Science. Has an endowed chair. What's not down here is that he's also a member of the National Academy of Medicine, National Academy of Engineering, and that earlier this year he received from President Obama the nation's highest award for technology, the National Medal of Technology. So if anybody Googles him, you can see pictures of him with Barack Obama. And speaking of Barack Obama, I first met him through the second person who's on the phone with us on the webinar, which is Guillermo, who organized this. And Guillermo's had an amazing career, also now full professor at Northwestern. In fact, he introduced me to Barack Obama when we both got honorary degrees there at the Northwestern in 2006. And he's very proud of all the work he's done. And the last person I wanted to put on is Monty Criquet, and she actually was at Wellesley. She was one of my very first students. She was an undergraduate, and actually wrote a couple papers with us on how you could deliver insulin, and she's now a psychiatrist in North Carolina. But and it's been wonderful just to see how their careers, and really everyone's careers in our lab, has gone. There's a few other comments that I was asked to talk about, like how do I choose people? Well, we've been very fortunate that we get a lot of applicants in our lab, and I actually just do, if somebody gets to be a graduate student at MIT, well, that or an undergraduate, they've already gone through a tough selection process. But I then even look further, do people really want to do the kind of work we do? And the kind of work we do isn't just driven purely by curiosity, which I think we certainly have, but also by the desire to get this out to the real world and help patients. And so that would be one thing. And if somebody's a postdoc, I look tremendously at what their advisors say, how good they are. And I also, what's very important to me is that they not only be great scientists, but they be great human beings. And that's been very important to me as well. The next question is, how do I pick projects? And there, to me, I guess I keep looking like, how can we, with the skills that we have in the lab, the people we have, my own skills, how can we make the greatest impact on the world? And so that's generally the guide that I've used and what we've tried to do. Right now, we're actually doing an enormous amount of work with the Gates Foundation. I like to think that we've helped people in the first world a lot, but we're now trying to see if we can use the types of things that we've done in drug delivery and other areas to help, really the billions of people in the third world. And the last question about how do I resolve conflict? I probably should ask Guillermo to answer that for me. I'm not sure that I'm necessarily do anything perfect. I guess the first thing is to not have conflict in the first place. When I mentioned about great people, it's interesting. Early on in my career, I did have some conflict with not me personally, but there was conflict in the lab because some people didn't get along. And then you'd have to figure out how to move certain people into places where they couldn't interact with other people. But the best way to resolve conflict is to make sure it doesn't happen in the first place. So we do a lot of due diligence on people before they come and how do they get along with others. And that's why I said it really means a great deal to me what their advisors say. I might interview somebody for half an hour to an hour, but I'm not that great at telling how smart they are or how nice a person they are. But I'd like to think that somebody they work with for 4 or 5 years would be able to. So I rely heavily on what they say, and that's actually something we even ask in questions, either verbally or in written form, about how well people interact. So the number one rule is try not to have conflict. I guess the number two rule is if it does occur, to me, I think it's very important to be very open about it. I've certainly worked with people who are sort of passive-aggressive. They don't like to deal with conflict. To me, I think if there's a problem, I'd rather get people together, and even if it's a little bit unpleasant, try to stop it before it gets bad. So, because conflict can happen. So I don't know that what I've said there is terribly profound, but that's what we do in terms of conflict. So what I thought I'd do is actually stop here and really, there are a number of questions. I'd be happy to answer whatever people want to ask, and I'll do the best I can. Thank you so much for having me.
Thank you very, very much for that very insightful discussion about your early days and how you've done so well throughout the years. Very, very inspiring, Bob. What I'd like to do now is to take the time to get your questions, the questions from the audience. You may continue to submit questions using the online chat feature on your screen. If you can send those to me, please. I'll relay those to Dr. Langer. And I hope to get as many as I can in before the webinar ends. And let's see here. I haven't seen any yet. Okay, here we go. 1 second. So, Bob, I have a question here from Erica Moore. What advice would you offer for graduate students unsure about pursuing academia? There's a lot of uncertainty nowadays about, the academic research pathway. So what advice would you give grad students about this?
Well, I think a couple of things. If you want to pursue academia today, I think, first, I'm a big believer that it's more important to have great publications and top impact journals than it is to have a lot of publications. I think the second thing I'd say is it's very important in today's world to learn how to write grants. That really is a significant part of what a young faculty member would do. And one of the things, as Guillermo knows, is like partly because our lab is big, but partly because I also think it's good experience, is I've always had the graduate students and postdocs help us on grants. And a third thing I'd say is I think if you're a graduate student and you want to go into academia, I also think it's very important to probably do some type of postdoc. That wasn't necessarily true when I was a graduate student, but that was many years ago. I think today having a postdoc is really helpful because you'll get more papers, you'll get hopefully some grant writing experience. Like I'd always recommend to people to write postdoctoral fellowships because you can learn a lot from that even if you don't get them. And also it'll give you a running start when you start to become a faculty member, and you'll have more ideas. So I think that those are some of the advice that I'd give.
Great, thank you. Second question is, were you able to continue your teaching and outreach activities after you started your own lab at MIT?
You know, I did some, and I still actually give quite a few talks to high schools, and we actually have high schools even come here to the lab, you know, where myself and other people, in fact, ILDA organizes those too, give talks. But I don't do as much as I used to. But we continue to do some, and we've had everybody from fifth graders here to, you know, high school students, and you know, and then I've gone out in the community and given lectures to a number of high schools as well.
Oh, excellent, excellent. Another question. What advice would you give an undergraduate student trying to decide between going into research versus joining a job in industry?
Well, of course, they may be the same. I mean, you know, so I think it depends. I would say a lot of times a job in industry could be a research job. And I don't know that if the question is what to do right after you graduate, my feeling is I don't know that what you do for the first couple of years after you graduate determines your whole life. So I think you actually have a lot of flexibility. My feeling is you could work in industry. I think that's fine, and I think you'll get a feel for what industry's like. You could go to graduate school, but I don't think that. I think the most important thing I think is to keep options open, so that and also, the real key to me is to follow your passion. Whatever you feel you love, that's what you should do. You shouldn't do it for money. You shouldn't do it because your parents tell you to do it. You should do it because you really feel in your heart you love it.
Gotcha. Very, very nice. We have a question from Wembo Gong. This is, he says, international students face visa issues when entering industry, and he would like to know if you have any advice to suggest for international students, which we've had a lot in our labs in the past. How do they go about finding jobs or careers in industry? Is it the same as it used to be back in the earlier days, or is it a lot more difficult to take international students?
My experience has been companies do take international students frequently. I think my sense is that the companies will take the best athletes, so to speak. They want to take the best students. And, you know, and again, if they're really tiny and can't afford, you know, say a lawyer for the individual, then that might be something to consider. But most of them, you know, have enough money, and what they really want is to hire great people. So I think that being a great person would be the most important thing. If they feel you're the right person for the job, I think that's what will really determine whether you'll get the job or not.
Gotcha. I have one question from one of our committee members. He would like to know what are your criteria for identifying underrepresented groups, specifically race, gender, geographical setting, maybe socio-economical factors. But he'd like to know if you, when you try to make these decisions, in addition to the best and the brightest, do these other identifiers come into your mind when making that decision?
I would say probably not. I mean, we have had plenty, as you know, of people of all types from many, many countries, from many, many, you know, women, men, you know, different minorities. But I really just look for the things that I mentioned. I look for the passion. I look for, you know, the person being a good person and the person being a good scientist. You know, I don't really look for, I mean, every so often, I guess, you know, when we've had, I want to make sure, because we have a softball team, that the person is a very good softball player. But other than that, probably nothing is key.
Gotcha. Thank you very much. I have one here. Your research is highly translational. You've been very successful at eventually placing products in the market. How do you balance a depth of knowledge with breadth, with the range of applications that you've explored over the years? And what is your advice to a new assistant professor that's trying to get started? Oftentimes they tell the assistant professor, you have to focus, but what is your advice in that matter?
Yeah, well, my advice is to do depth first. I think that whatever you do, you want to do a great job on, and it's better to do one thing well than 4 things fair, or even 10 things. So I really do feel that depth is the most important thing. Once you've really done one thing well, then maybe you can try to do 2. But doing one thing well is hard enough. And that would be part of the advice for assistant professors is to do one thing well, and it can be translational work, it can be basic work. It should just be something that you love, and that's also part of my advice to people who are new assistant professors, is to pick something that you feel is important, that you have a passion for, and then you really go after that particular area and try to. But I'd also pick things that you feel can have a high impact. I think if you do that, that'll certainly help you advance to higher level professorships and everything else with it.
Yeah, well said, Bob, well said.
And I should add, Guillermo, and maybe this is obvious from what I said before, you want to be able to raise money. You have to make that important. You want to get good at writing grants or, writing patents or whatever it is that will help bring money, because if you can have money, that gives you the flexibility to hire just terrific people.
That's right. That's well said. Another question, what is your advice to basic scientists or basic bioengineers who are not necessarily making something such as a new polymer and may have a more difficult time in this funding climate? For example, maybe computational, people with computational backgrounds or interests. What would you say to them?
You mean, let me make sure I understand the question. What would I say to people
who are pursuing basic science or the basic science in the bioengineering area that are not necessarily not applied scientists, maybe not polymer?
Yeah, I'd really say the same kind of thing. I think that the most important thing is to do what you love. I think basic, we do a lot of basic science in the lab, published in Science or Nature and places like that. And I think it's good to do basic science. We try to do both good basic science and good applied science, but I think basic science is terrific. I mean, that's where some unbelievable discoveries have come from, and so I think that's just terrific, and I would encourage that. I think, but that is something, if you do basic science, it's important to learn how to present it in a way in grants that people will say, boy, I really want to fund this, because you will need the money to develop these things, even when it's basic.
Gotcha. So this is a question from me. If you had to guess, how many countries have been represented through your lab over the years? Have you ever done that number? Maybe Ilda would know.
Ilda may know better. I'm sure it's over 30, but I don't know if it's over 40 or what.
At least 30, I would say. I agree with that.
Yeah, Ilda's thinking as you said that, but I mean, we have, we've Panama, we've had almost every company. Yeah, she said pretty much every country in Europe. We've had people from Russia. We've had Australia, Japan,
Africa
Korea,
as well.
China.
Yeah.
Africa, yes, yeah. Actually, you're right. Chris Alabi was, and others. So yeah, maybe it's over 50. I mean, it's a lot. That's a good exercise. We'll have to do that.
That'd be good to.
But it's a huge number.
I agree. Let's see. We have another question coming in here.
Iran, Israel. Yes.
We have about 5 or 10 more minutes or so. You pointed out earlier in your career that people were, other scientists or professors were giving you a fairly hard time about your radical, quote-unquote radical, new ideas at the time about proteins diffusing through polymers. What kept you going, that persistence that people say you typically have, whether or not it's as an assistant professor or as an entrepreneur trying to start a new company? There's this quality that you have to have, right, to keep going. Can you perhaps share how do you manage to, part of it you said you did not get to read some of the comments about these things not being possible, so I'm sure that helps. But is there anything else you could add to that as far as what techniques or what about your personality allowed you to pursue and keep going until you're able to find that 1 out of 200 or 1 out of 300 molecular experiment that worked and eventually build a field out of it?
I think maybe 2 things. 1, being very stubborn, and 2, just believing in what we were doing.
No, that's good point. Stubborn makes sense.
Yeah,
Right.
very stubborn.
Excellent, excellent. So, well, Bob, I don't see there aren't any more questions coming through. So what I'd like to do is thank you once again for taking this time out of your schedule to join us in this endeavor, and maybe some closing remarks. Given the climate in the country right now, this divisive elections and so forth, what do you see as the future for BMES as far as we're trying to be inclusive, get more underrepresented minorities, more women into our field? We're trying to broadcast what we're all about to the rest of the world. As you know, biomedical engineering is a fairly new discipline as far as engineering. What advice would you give us, or would you give to those students and future faculty that would like to pursue biomedical engineer and feel that they have a place in academia and research and in getting grants? Any encouraging words on your part?
Well, I'm a huge believer in the field. I think biomedical engineering is tremendous. I mean, one thing we can do is just look at the number of people who were involved in it 20, 30 years ago and the number of people who are involved in it today. And it's just astounding what's happened, in terms of the increase. It's also been astounding in terms of the number of new departments that have come up. I don't know that any field has done quite as well as biomedical engineering in terms of new academic departments. I'd say the same for people in companies. It's been an amazing time with the biotech industry. So I think it's a great field. I'm obviously prejudiced. I think it's a great field for a couple reasons. One, I think you can do some really good science and engineering. But two, I think you can fundamentally change people's lives. One of the things that I think has been very rewarding for the people who work in our lab is seeing the direct effect you can have on patients. And I think that's been really exciting, and I think anybody that participates in biomedical engineering has the potential to just make a huge impact on mankind. In terms of politics, what I've watched over the years with NIH funding and other funding, there's ups and downs. I was even saying to my wife last night that, what happens is usually people win elections, there's a shift from one side to the other, and then it goes back the other way because people get fed up with the people, that are in office. And I think that or the types of people, and I think that keeps happening. And I expect will happen again. So I think that it's kind of like an internal checks and balances, or at least it's been that way so far in the 40-some years I've done. So sometimes it's harder to get money from the government, but when it's harder to get money from government, then, that might be a time when you might be able to get more funds from industry. And the other, of course, big thing that's happening today for people who want to get funding is foundations. There are a lot of people who've made a significant amount of money, and they have causes in, say, diabetes or world health and or cancer, and they want to fund these things. And so I think that one just has to be alert to what happens and, in terms of what's going on in society. But I think that there's certainly funding out there, though that's a skill set that it's worthwhile developing.
Excellent. Well, Bob, thank you very much, and thank you, the audience, for your questions. This is all the time we have for today. There are no more questions coming through. And for those of you who did not get or were a bit shy to ask questions, this event has been recorded, so you can have access to it, as well as perhaps follow-up questions that you can send, and the speaker can also answer those questions afterwards. This concludes our webinar, and again, thank you for joining us, and please stay online to take a few minutes to complete the brief webinar evaluation before you go. I would love to get your feedback, and once again, thank you for taking the time. And thanks, Bob, to you again for being an excellent advisor back then and a friend today.
Thank you, Guillermo, and for everyone for listening. It's been a pleasure to speak to you, and I wish you all a lot of luck.
Thank you. We hope you will join us again at some point in the future for other BMES events. Thank you all.